Welcome to the afternoon symposium. So it's my honor to introduce the academician Oh Jingxiong, James Oh. And Dr. Oh received his bachelor degree from National Taiwan University at Department of Zoology, which unfortunately does not exist anymore. And then he did his PhD at Caltech, and then postdoc at UCSF. And then afterwards, he went to University of Southern California, became a faculty there, and then promoted to associate professor and then full professor. So he is now a professor at Department of Microbiology and Immunology at University of Southern California. And Dr. Oh's research interests majorly on the understanding the biology of hepatitis B virus and hepatitis C virus to understand how they cause the cancer and liver diseases. And he recently has been more interested in the development of cancer in liver. And so he has made a tremendous amount of contribution in this area. So let's welcome Dr. Oh to hear about his work.
Okay, thank you, Susan, for the kind introduction. I'm a virologist, so it's very unusual for me to go out and give a talk without mentioning viruses. And today is going to be one of those rare exceptions. I've been very interested in studying liver cancer, like Susan mentioned, that because the hepatitis B virus and hepatitis C virus are the two major causes of liver cancer. A few years ago, I became interested in the relationship between autophagy and liver cancer, because at that time, the role of autophagy in carcinogenesis was very controversial. Some reports say that the autophagy is anti-tumorigenic, but others argue that the autophagy is pro-tumorigenic. So we decided to look at the relationship between autophagy and the liver cancer. And just in case you are not familiar with the autophagy, I briefly explained it to you. Autophagy plays a very important role in maintaining cellular homeostasis, and it's ongoing in the cells all the time. And the autophagy begins with the formation of the membrane crescent. It's called the phagophore. The membrane of phagophore will then expand to form an enclosed double membrane vesicle called the autophagosomes. Autophagosomes will mature by fusing with lysosomes to form autolysosomes. And the cargoes of the autophagosomes will then be digested by lysosomal enzymes for recycling. And as I mentioned, the autophagy is very important for maintaining cellular homeostasis because it's used by the cells to remove the protein aggregates and the damaged organelles like mitochondria and ribosomes. So to understand the role of the autophagy in the hepatocarcinogenesis, what we did was to create a liver-specific knockout of ATG5. What we did was to cross-flux ATG5, and this is a gene essential for the formation of phagophore, and cross it to albumin Cree mice, and that led to the hepatocyte-specific knockout of the ATG5. So this is the Western blot analysis of the proteins in the liver. And this is the wild-type mice, and these are the ATG5 knockout mice. And you can see the knockout was very efficient. We detected very low level, very little ATG5. And this is the LC3 Western blot. LC3 is a cytosolic protein, but during autophagy, it becomes lipidated. And the lipidated form migrates faster on the gel. Okay? And you can see this lipidated form largely disappeared. And we also analyzed P62. P62 is a protein degraded by autophagy. So if autophagy is impaired, its level will go up. Okay, and that was indeed the case. So this result indicates that we had the ATG5 knockout was very efficient in our mice, and there the autophagy was impaired. So we then look at the possible liver tumor development in our knockout mice. And this is the red curve here is the ATG5 knockout mice. And you can see by 6 months of age, 20% of the mice will develop liver tumors. And this percentage goes to about 50% at 8 months of age. And then by 10 months of age, all the ATG5 knockout mice develop liver tumors. Now, if we look at the control mice, the wild-type mice, they did not develop liver tumors. until at about 12 months of age, when a single mouse developed a single tumor nodule. Okay, so the ATG5 knockout will lead to the development of liver tumors. So this result is consistent with the anti-tumorigenic function of autophagy, because when autophagy was impaired, liver tumor developed. And this is what the liver tumors look like. They are usually multifocal and they are very heterogeneous in size. So to understand why the ATG5 knockout would lead to the development of the liver tumors, we conducted electron microscopy to look at the mouse hepatocytes. And the one obvious difference, this is the wild-type mouse hepatocyte, and this is a knockout hepatocyte. And one obvious difference you can see here is the enlargement of the mitochondrial size. Okay? And this suggested that the mitochondrial physiology might be altered when the autophagy is impaired. And that was indeed the case. And this affects analysis of the mitochondrial membrane potential. And you can see for the knockout mice, comparing with the wild-type mice, the mitochondrial membrane potential has greatly reduced for a majority of the mitochondria in the knockout mice. Okay? And the dysfunctional mitochondria can lead to the production of reactive oxygen species. Okay? And so we also measure the ROS in the mouse liver, and indeed the knockout mice, they have a higher level of ROS. Okay? I'm not showing you those results because of time constraint. But the oxidative stress can lead to oxidative DNA damage. Okay? So in this case, we measure the 8-hydroxydeoxyguanosine level. This is the marker of oxidative DNA damage. And we use the wild-type mouse liver and the spleen of the knockout mice as the controls. And you can see this is the liver of the knockout mice, the red bars. You can see there's an increase of the oxidative DNA damage. And the level peaks at about 6 months of age and remain at a very high level. And we also look at the gamma H2AX. It's induced by double-stranded DNA breaks. So it's a marker for DNA damage. And you can see for the wild-type mice, there's very few, we could not detect actually a gamma H2AX positive hepatocytes. But the knockout mice, you can easily see this gamma H2AX positive hepatocytes. So it becomes very clear to us why autophagy is anti-tumorigenic, because it's required to remove the dysfunctional mitochondria. In the absence of the autophagy, dysfunctional mitochondria will accumulate. They will lead to oxidative stress and oxidative DNA damage, and that's why liver tumor develops. Okay, and of course we can confirm that result by feeding mice with the antioxidant N-acetylcysteine. Okay, this is the antioxidant. And you can see the mice fed with the knockout mice fed with the NAC. You can see there's a reduction of liver tumor incidence at both 8 months and 10 months of age. If we look at the tumor burdens by counting the tumor nodules in those mice that develop liver tumors, you can see the total number of liver tumors were also reduced. And if we count the tumor size, it was also reduced. So the overall tumor burden was reduced. So the antioxidant can reduce the liver tumor incidence. It was also after seeing these results, they have become convinced that antioxidant is actually good for us. It will reduce the cancer possibility. Okay, one thing that I have not mentioned is that even though these knockout mice develop liver tumors at very high frequency, when we did the histological analysis, we found that all those tumors, they were benign focal hyperplasia or some adenomas. In other words, they are benign liver tumors. We could not find any hepatocellular carcinoma. Okay, and so it was very curious. These mice, when autophagy is impaired, they would develop liver tumors at very high frequency, but none of them are the malignant hepatocellular carcinoma. So to understand why, we treated the mice with DEN diethylnitrosamine. It's a common dietary carcinogen. And you can see that for the knockout mice, first let's look at the wild-type mice. Okay, you can see that by 6 months of age, about 60% of the wild-type mice, they would develop liver tumors after the DEN treatment. And by 8 months of age, 100% of the wild-type mice would develop liver tumors. But when we look at the knockout mice, at 4 months of age, already 80% of them will develop liver tumors after they were treated with DEN. And by 6 months of age, 100% of them will develop liver tumors. Okay. Now, without DEN treatment, the knockout mice, it took them 6 months to develop liver tumors, but now it took only 4 months. So DEN does facilitate the development of liver tumors. We then did the histological analysis again. And this is a knockout mice. Again, we could find only benign liver tumors. We could not find hepatocellular carcinoma, not even after DEN treatment. But for the wild-type mice, we can easily detect hepatocellular carcinoma. Okay, you can see that the histology is very different between benign tumors and malignant SCC. Okay. But we also did another experiment just to confirm the results. This time we did the Western blot. Glypican-3 is a marker of SCC. Okay, so we randomly picked tumors from different wild-type mice that have been treated with DEN, and this is a non-tumor liver tissue as a control. You can see the glypican expression level varies a lot between different tumors. Some of them are very high. These are the hepatomas, SCC. And some of them are very low, and these are probably the benign tumors, okay, in the process of becoming a malignant SCC. But when we look at the knockout mice, again, this is a non-tumor tissue as a control. You can see that for the knockout mice, the tumors, the glypican-3 level, they were all very low, okay? So confirming that these are all the benign tumors. They are not hepatocellular carcinoma. Otherwise, the glypican-3 level would be high. Okay, so the question is that why without autophagy, these tumors cannot progress to become malignant SCC. So just to summarize what I have told you so far. Now, what I have demonstrated to you is that autophagy has dual effect in hepatocarcinogenesis. It's anti-tumorigenic because it needs to remove dysfunctional mitochondria to minimize oxidative stress and oxidative DNA damage. Okay, but without the autophagy, then the benign tumor cannot progress to become malignant hepatoma. So in that sense, it's pertumorigenic. You need autophagy for benign tumors to become malignant hepatoma. Okay. The question is why? Why the autophagy is required for the benign tumor to become malignant hepatocellular carcinoma? There are several reasons. We have done extensive studies to understand the process. But one of the most important reasons that we think is due to the loss of cancer stem cells. Okay? Here, just to show you, this is a tumor isolated from wild-type mice that have been treated with DEN. Okay, here we measure the CD49F and CD133. These are the two markers of stem cells. Okay, so you can see that in this tumor, about 0.85% of the tumor cells are CD49F and CD133 double positive. Okay, these are presumably the cancer stem cells because they display the stem cell markers. But when we look at the tumor isolated from the knockout mice, and you can see this percentage was reduced to 0.14%. They pretty much all disappeared. There's a 6-fold reduction, suggesting that autophagy may be required to maintain cancer stem cells. Without it, cancer stem cell population will be lost. Okay. So to understand whether autophagy is indeed required for maintaining cancer stem cells, we shifted our attention to HepG2 cells. It's a human hepatoblastoma cell line. It's easier to understand the mechanism using cell cultures. And here, HepG2 cells, we did FACS analysis. About 6% of them are CD133 positive. The remaining HepG2 cells are CD133 negative. And if we treated the HepG2 cells with 3-methyladenine, or beflomycin, both of them are the inhibitors of autophagy. And you can see that when autophagy was inhibited, the CD133, cancer stem-like cells population will be reduced. But when we treated the cells with rapamycin or serum-free media, these are the autophagy inducers. And you can see the CD133 positive cells population goes up. Okay, so the as ATG5 knockout had the same result. It reduces the CD133 positive cells. So this result is consistent with the in vivo results. The autophagy actually is required for maintaining cancer stem cells. When autophagy is suppressed, cancer stem cell population drops. When autophagy is enhanced, cancer stem cell population goes up. And we also conducted the Western blot analysis to verify the result. And first, let's look at the P62, okay? And we did treated the cells with the ATG5 shRNA, O3MA, and beflormycin. All three of them inhibits mitophagy. You can see P62 level goes up because this is a protein degraded by autophagy. Okay, so they indeed suppresses autophagy. That's why P62 level went up. If we treated the cells with rapamycin and serine-free media to induce autophagy, then you can see P62 signal is lost because it's removed by autophagy. Okay, and then we look at the CD133 expression level. You can see the autophagy inhibitors reduce the CD133 protein level, but the autophagy inducers increase CD133 protein level. We also look at the NALOG. NALOG is a transcription factor, very important for the self-renewal of stem cells. Okay, and you can see the autophagy inhibitors cause the NALOG level to become undetectable, but the autophagy inducers will increase the NALOG protein levels. We also look at the OX4 and SOX2. These are the two transcription factors that activate the expression of NALOG, and they were not affected by the autophagy inducers or inhibitors. So we know that the step of the autophagy on the regulation of cancer stem cells is at a step between OX4, SOX2, and NALOG. It's upstream of NALOG, but probably downstream of SOX2 and OX4. I kept calling these CD133 positive cells cancer stem cells. And what's the evidence for that? Okay, there are two major assays that are used frequently to analyze cancer stem cells. One is called this self-renewal assay, or the sphere formation assay. In this case, we took CD133 positive HepG2 cells and plated them out on a low attachment Petri dish. And you can see the cells will proliferate and grow and to form like cell clumps. We call this a sphere formation assay or spheroid formation assay. But if we took CD133 minus cells and plated it out on the Petri dish, they will not grow because they cannot self-renew. Okay, so this is one criterion of the cancer stem cells. The other one is to tumor formation in the NSG mice or the immunodeficient NUT mice. Okay, and I will show you the result later. CD133 positive cells can also form tumors, but not CD133 negative cells. Okay, so these are indeed, they have the properties of cancer stem cells. So here you can see the if we treated the cells with knockdown the ATG5, you can see the self-renewal ability of these CD133 cells will be reduced. Okay, and CD133 minus cells cannot efficiently form spheres. And the same thing, if we inhibited autophagy with 3-MA or beflormycin, the sphere forming ability will be reduced. But if we induce autophagy with rapamycin or serine-free media, you can see the self-renewal ability of the stem cells went up. Okay. Okay. That result was obtained with HepG2 cells. Okay, so after we got those results, I told my postdoc saying that, well, you should look at other tumor cell lines also because we want to make sure that the result is not specific to HepG2 cells. So my postdoc analyzed Hep3B cells and HU7 cells. These are two different human hepatoma cell lines. And after he had done the experiments, he came to me and he was very disappointed because what he found was that these are the CD133 positive cells. These are the Hep3B cells. And he found the autophagy inducers, those inhibitors, they had no effect on the CD133 positive cell populations and for Hep3B cells. For HU7 cells, he also could not detect any effect. Autophagy inducers or inhibitors had no effect on the total population of CD133 positive cells. So he was very disappointed. And I have been working with these cell lines, HepG2, Hep3B, and HU7 cells for 30 years. So I know there's one fundamental difference between these three different cell lines. That's P53 tumor suppressor. HepG2 cells express the wild type P53. Hep3B cells did not express P53. And HU7 cells express the defective P53. So I told my postdoc, and I tried to comfort him and told him that don't get too disappointed yet. Let's see whether if you put P53 back into these two cell lines, what will happen, okay? So he did an experiment, and this is what happens. He put P53 into HEP3B cells. Now, the total CD133 positive cells, the population was reduced from 20% to about 8%, okay? But now take this P53 positive HEP3B cells. He treated with the 3-MA to inhibit autophagy. Now, the CD133 positive cells, the number got reduced. But when you enhance autophagy, then this number goes up. So he did the same experiment with HU7 cells by expressing P53 in HU7 cells. Now the cells also responded to the autophagy inhibitors and inducers. So it was really the P53 that's required for autophagy to exert its effect on cancer stem cells. Okay? So just to summarize again what I just told you, autophagy positively regulates cancer stem cells. When it's impaired, cancer stem cell population drops. When it's enhanced, cancer stem cell population goes up. But this process is P53 dependent. So the question is that how does P53 mediate the effect of autophagy on cancer stem cells? Okay, so we first look at the effect of autophagy on P53. And you can see these autophagy inhibitors, including ATG5 knockdown. You can see the P53 level will go up. And also the P53 phosphorylated at serine 392, the level will also go up. But when we use the autophagy inducers, then you can see P53 level will drop. And this phosphorylation at serine 392 also will disappear. Okay? So we know autophagy can regulate P53. Okay, it can reduce the expression level of P53, and it can suppress P53 phosphorylation at serine 392. Okay, so we know autophagy can negatively regulate P53. So how does P53 regulate cancer stem cells? Okay, so for that we focus our attention on the relationship between P53 and NALOG. NALOG, as I said, is a very important transcription factor, essential for the cell. That's very important for the self-renewal of these cancer stem cells. So I've shown you that autophagy can positively regulate NALOG protein level in the Western blot analysis. Okay, and so we also look at the RNA, and you can see autophagy inhibition led to reduction of the NALOG RNA, but autophagy induction increased the NALOG RNA level. Okay, so the effect of autophagy on NALOG is at the transcriptional level. So we look at the possible effect of P53 on NALOG RNA transcription. This is a NALOG promoter. Okay, so we link it to the luciferase reporter and look at the possible effect of P53 on NALOG promoter. And you can see if we co-transfected this reporter plasmid with P53 expression plasmid into the HEP3B cells, and you can see the NALOG promoter activity will be suppressed. So P53 can indeed negatively regulate the NALOG promoter activity. And so we look at the NALOG promoter, we could find a possible P53 response element. And indeed, when we did the deletion mapping experiment, this region is the sequence that's responsive to P53 suppression. So we took this DNA fragment and used it as a probe to confirm that whether P53 can indeed bind to it. So they would do the electrophoretic mobility gel shift assay. And so in this case, we incubate the DNA probe with HepG2 nuclear extracts. And you can see it will cause a band shift on the gel. And when we also incubate this with the anti-P53 antibody that recognize phosphoserine 392, then you can see we can get a super shifted band. Okay, and if we use enough antibody, all the band will be shifted. So this result confirmed that this site is indeed the P53 binding site. P53 binds to it to suppress the NALOG promoter. Okay, and more importantly, all the P53 binds to this band, binds to this sequence is phosphorylated at serine 392 because this antibody recognizes phosphoserine 392, and all the bands could be super shifted. So this P53 is phosphorylated at serine 392. phosphorylation at serine 392 will activate the nuclear translocation of p53 and also activates this DNA binding activity. Okay, this is a very important phosphorylation event for regulating p53 activity. Okay. So what I have shown you is that p53 can negatively regulate p53. The autophagy can negatively regulate p53 and its phosphorylation and serine 392, and that will suppress the expression because p53 will suppress NALOG. So this downregulation will lead to the activation of NALOG and increase the cancer stem cell population. Okay, so this is what I have shown you. Okay, so now we will come back to the question. I have shown you that autophagy can negatively regulate p53. So how does autophagy do that? Okay. For that, I need to mention mitophagy. Mitophagy is the selective removal of mitochondria by autophagy. Okay. There are two drugs that are commonly used to study mitophagy. One is called MDV1. The other one is called CCCP. MDV1 is the mitophagy inhibitor. CCCP is the mitophagy inducer. Okay? And here, this is the Western blot. We use TOM20 and TIM23 as markers for mitochondria. TOM20 is the outer membrane protein of mitochondria. TIM23 is the inner membrane protein of mitochondria. So this is the total cell lysis for Western blot. If we treated HepG2 cells with MDV1 to inhibit mitophagy, you can see the level of TOM20 and TIM23 went up. This indicates there's an increase of mitochondria amount. Okay, so it inhibits mitophagy. But if we treated cells with CCCP, you can see TOM20 and TIM23 mitochondria is lost. Okay, so it confirms the activity of these two drugs. One inhibits mitophagy and one enhances mitophagy. And here is the Western blot of LC32. Okay, now in this case, we purify mitochondria from the cells, control cells, or the cells that have been treated with MDV1 or CCCP. Okay, then we look at the first. Let's look at the CCCP. Okay, the mitochondria. You can see we can detect a lot of this lipidated LC3, meaning that this mitochondria in the cells that have been treated with CCCP are associated with autophagosomes. That's why there's a lot of lipidated LC3 there. But if we treated cells with MDV1 that inhibits mitophagy, you see the mitochondrial fraction. There's no lipidated LC3. So the mitochondria in cells treated with MDV1 is not associated with autophagosomes. Okay, so this basically just a result, a control experiment to show you that these two drugs can indeed inhibit or induces mitophagy. Okay. So we then look at the effect of mitophagy on cancer stem cells. Now again, we look at the CD133 cells. And you can see if we treated the cells with the mitophagy inhibitor MDV1, you can see that it reduced the CD133 positive cells population. Okay? But if we treated the cells with CCCP, the mitophagy inducer, you can see the cancer stem cell population went up. Okay? We also look at the self-renewal ability of the cancer stem cells by conducting the sphere formation assay. And you can see again, mitophagy inhibitor reduce the self-renewal ability of the cancer stem cells, but CCCP increased it. Okay, and we also did a Western blot. Again, this is the mitophagy inducers increase CD133 level and increase NALOG, reduces p53 and p53 phosphorylation at serine 392. But mitophagy inhibitors reduce the CD133 NALOG and increase p53 and p53 phosphorylation at serine 392. So whatever we were seeing with autophagy on cancer stem cells, we could reproduce it with mitophagy. Okay, so it looks like the effect of autophagy on cancer stem cells is actually going through mitophagy, because by manipulating mitophagy alone was sufficient to regulate cancer stem cells. Okay, so we can now narrow our focus to mitophagy. Okay, so now the next step is to understand the same as autophagy. Mitophagy can downregulate p53 and its phosphorylation at serine 392 and increase the expression of NALOG and cancer stem cells. Okay, So how does mitophagy negatively regulate p53 and its phosphorylation at serine 392? So we pushed our study a step further. It's mitophagy. So how does mitophagy regulate p53? Okay, to do that, we look at the relationship between mitochondria and p53 that's phosphorylated as serine 392, the activated p53. Okay, we did a confocal microscopy. Okay, now we stain the p53 with the activated p53 in red color. Okay, and the green color here we stain the TOM20 as a marker for mitochondria. Then we look at the relationship between the activated p53 and the mitochondria. Okay, and here let's look at the enlarged picture over here. And you can see that in control cells treated with DMSO, the activated p53, they are localized primarily to the nucleus. Okay, and there's some also you can detect them in the cytoplasm. And for the MDV1 treated cells, this inhibits mitophagy, and you can see there seems to be an increase of nuclear p53. Okay, the activated p53. And but most interestingly is this one. The cells treated with CCCP, the mitophagy inducer. Now we could not detect the activated p53 in the nucleus. Instead, they are all in the cytoplasm. And actually they co-localize with mitochondria, or they are in the vicinity of mitochondria. Now for this particular experiment, we treated cells with CCCP for only a short period of time. Otherwise, all p53 and mitochondria will disappear, making it very difficult to do the study. That's why we treated it for only a short period of time. And now in this case, p53 is localized to mitochondria. So this makes a lot of sense to us now that the way mitochondria, mitophagy, the way mitophagy negatively regulate p53 is because when mitophagy is induced, p53 is localized to mitochondria. And because mitophagy removes mitochondria, so p53 is actually removed together with mitochondria by mitophagy. That's why mitophagy negatively regulates p53 and its phosphorylation at serine 392. That's what we think. We confirmed the confocal microscopy results with subserial fractionation analysis. Okay, and this is the mitochondrial fraction, this is the cytosolic fraction, and this is the nuclear fraction. And you can see the cells treated with the CCCP, the mitophagy inducer. You can see the p53 is indeed associated with mitochondria, and very low level of them are found elsewhere. Okay, but you treated cells with MDV1, the mitophagy inhibitor, then you can see the p53, the activated p53 is localized to the nucleus. Okay. So, how does mitophagy regulate the subcellular localization of p53? We had to push this question, keep pushing it to understand the whole process. And here, I need to introduce to you this kinase called PINK1. PINK1 is P10-induced kinase. This kinase plays very important role in mitophagy. So PINK1, after it's synthesized, it has a mitochondrial targeting signal. It will be localized to mitochondria. Okay? And its N-terminus will be truncated by a protease located in the inner membrane of mitochondria. And the N-terminally truncated PINK1 will then be released back into the cytosol and will be degraded by proteasomes. It's a very labile protein. Okay? But when mitochondria is damaged, depolarized, PINK1 will be localized to mitochondria, but it will no longer be cleaved at the N-terminus. Okay? So once it's stabilized on mitochondria, it will recruit parking and activate parking. Parking is the E3 ubiquitinase. E3 ubiquitin protein. Okay, ubiquitinase. Okay, so after recruiting the parking and activates it, parking will ubiquitinate the mitochondria surface proteins. Okay, and this ubiquitination will trigger the mitophagy. Autophagosomes will form on mitochondria, and the mitochondria will be removed by mitophagy. Because PINK1 is very important for mitophagy, so I told my postdoc and said, well, to understand the effect of mitophagy on p53, why don't you look at PINK1? Okay? Why don't you knock down PINK1 or overexpress PINK1 and see what happens to p53? Okay? That turned out to be a very naive suggestion to my postdoc. I was very embarrassed, actually. Because This is what he found. He knocked down PINK1. There's no effect on TOM20. This is the marker for mitochondria. When you overexpress PINK1, there's no effect on mitochondria either. And it's because I was very naive, because PINK1, although it's very important for mitophagy, is not essential. There are other pathways for mitophagy. So knocking down PINK1, it did not affect basal mitophagy. That's why there's no effect on TOM20 mitochondria. Overexpressing it is not going to increase the number of depolarized mitochondria. That's why it had no effect on mitophagy. Okay? And then you look at P53, there's no effect on overall level of P53, whether the PINK1 was knocked down or overexpressed. But what struck me was that when my postdoc knocked down PINK1, the P53 phosphorylation at serum 392 was reduced. When he overexpressed it, P53 phosphorylation at serum 392 was increased. I got very excited at that time, actually. I told my postdoc, you know, people have been looking for this kinase for 25 years. They phosphorylate P52 at serum 392. You might actually have discovered it. You might have found this kinase, okay? Well, of course, he also did the CD132 and NADOC because P53 was reduced, and that's why CD132 went up, NADOC went up. And in this case, P53 was activated, and that's why NADOC went down and the CD132 went down. So I told my postdoc to verify whether PINK1 is indeed a kinase for P53, serum 392. And so he did this experiment. He did the knockdown experiments. Over the past 25 years, several kinases have been reported to be the kinase that phosphorylate P53 at serum 392. And this includes the casein kinase 2, P38, the MEK kinase, and the double-stranded RNA-dependent protein kinase, PKR, and CDK9. And he did the knockdown experiments on every single one of them. None of them affected the phosphorylation of P53 at serum 392, with the exception of PINK1. When he knocked down PINK1, the phosphorylation of P53 is gone. And of course, he can also purify PINK1 from the HEP-G2, HEP3B, and Q7 cells, and did the in vitro phosphorylation assay. And then again, he can see that the P53 can be phosphorylated by PINK1 in vitro. Okay, so he confirmed that the PINK1 is indeed the kinase that phosphorylates P53. And he also did the co-IP experiments. And just to make the long story short, PINK1 actually can bind to P53 only when it's associated with mitochondria. Okay, not in the cytosolic fraction. There's no co-IP there. Okay, so this comes to the model. This is what we think is happening. Okay, in the healthy cell, PINK1 is localized transiently to the mitochondria, then removed and degraded. But during that process, probably a small amount of P53 will be captured and phosphorylated, but that amount is very small. It's insufficient to shut off the NADOC expression. Okay, so we have some cancer stem cells. But when mitophagy is enhanced, okay, PINK1 will bind to P53, phosphorylate it. But before P53 can escape, it's trapped inside autophagosomes and it's degraded. And in this case, ACH4 and SOX2 will activate NADOC expression to increase the cancer stem cells. Now, when mitophagy is impaired, depolarized mitochondria accumulates. So there's a lot of PINK1 are associated with the mitochondria. Then they will capture P53. But then there's no autophagy to remove P53. So P53 will be released, go into the nucleus, shut down NADOC expression. So there's a loss of cancer stem cells. So this is what we think is happening. Okay, finally, just to show you these additional slides, results. If PINK1 is so important for regulating P53 activity, so what would happen if we knock down PINK1 when autophagy was impaired? This is the inoculation of the CD133 positive HEP-G2 cells into the nude mice. You can see from tumors. But when we knock down ATG5 to impair the autophagy, the tumor cannot grow. Okay? But when we did a double knockdown PINK1, you see the tumorigenesis is restored. That's why I said that cancer stem cell is probably one of the most important reasons for facilitating tumorigenesis. Because even when autophagy was impaired, as long as we knock down PINK1, so that there is no P53 activation. Then the tumorigenesis will be restored. And of course, if we just knock down PINK1 alone, the tumor is even bigger because autophagy also has some other effects on the tumor growth. And this is just the Western blot, I think, because I'm running out of time, so I'll skip this one. So just come back to the original question. We started out to understand the role of autophagy in hepatocarcinogenesis. And what I've shown you is that autophagy actually had both anti-tumorigenic and pro-tumorigenic effect in the liver tumor growth. It's anti-tumorigenic because it's required to remove damaged mitochondria to minimize oxidative stress and DNA damage to prevent the liver tumor from developing. Okay? In the absence of it, this process will happen. There's oxidative DNA damage, then the tumor will grow. Okay? But then it will not progress to become the hepatocellular carcinoma because autophagy is also required to maintain cancer stem cells. Okay? Without cancer stem cells, the benign tumor cannot become malignant ones. So finally, the work actually I described today was done pretty much by only 2 postdocs. Yongjun Tian was the person who did the first half of the talk, the work. And Kai Liu Liu Kai was the person who did the cancer stem cells stories. I'll stop here. Thank you.